So today we will finish up, hopefully, our discussion on the spatial map formation of place cells in the hippocampus as preparation for these 3 talks next week at the IMA, and which you can join online. So we will finish discussing this paper on cell groups, reveal structure of stimulus space, published by Curto and Itzkov in 2008. And then we will also talk about some progress made by the Carlson group, published in 2012. They did somewhat similar things, but there's also some big differences as well, and we'll summarize some of those. Okay, so recall that we have place cells. So we've got these neurons over here. So let me get out my pen. So we've got our place cell, our neuron 1, 2, and 3, and it has a particular firing. Each of these cells have a particular firing pattern, and they depend upon where the mouse is. So for any place within this blue region, which is called the place field, corresponding to place cell 1, so then the blue place cell 1 will fire. When we enter into the green place field, then place cell 2 will fire. And when we enter into the red region, place cell 3 will fire. Now, keep in mind that all data is noisy, so there could easily be some extra firing over here, something's missing, and so data usually is quite noisy. But this is the basic idea. So in the past, what biologists have done is they have taken basically a rat or a mouse traveling in some space here, and they look at where the mouse is at particular times, and then they can correlate that with when the cell fires. So they basically will, you know, we've got our mouse, so this is my picture of a nice mouse. They can plant electrodes in various parts of the hippocampus to try to identify some, but not all, of the place cells. And then they see that, oh, as you're going around here, any place within this region here, you have basically a firing pattern, maybe a little bit of noise outside, but you have a firing pattern here, and so you note that since it's firing here when you're in this region, you associate being within this region, so this cell firing to being within this region here. That means that they can then, if they take a look at just the spiking data, they can then go back and say, well, if we take a look at just the spiking data, so this was the original path, but if you now ignore the original path and just look at the spiking data, you can approximate the path given that information. But the biologists were using the fact that they correlated the location of the place field to which place cell was firing. What the latest papers want to take a look at is just going straight from the firing to the topology, and later we'll talk about the geometry of the configuration space. So remember, we have our cell groups become our simplices. So each of our vertices represents a place cell. They could also represent a place field. We hope that the two are actually in agreement. But what we want is to get our simplicial complex from the firing information. And so if our edges correspond to the intersection of when two cells co-fire, we can get an edge between them. And we hope that that is in one-to-one correspondence with place fields, say 1 and 2, also intersecting. But remember that there is noise, and so while we want them to be an exact one-to-one correspondence, They're not an exact one-to-one correspondence, but we hope that the topology is still preserved. And that's basically what the 2008 paper looked at. And we know that if it is, that if we are taking a look at this down here, the place fields, then because our intersections are convex, we know that from our place fields, from our place fields by the nerve lemma, we do get the same homology as the actual environment. So we know that these two will give us the same topology. And the thing was, when you add noise to this, do you also get the same topology? And the 2000 paper said, yes, you do. Questions so far? So both papers, the 2008 and the 2012 paper, they both looked at can you recover the topology of the space by looking only at the spiking activity, by looking only at the place cells and not the actual environment that the mouse or the rat is traveling in. But in both papers, it is a proof of concept. So a later paper by Chen does analyze real experimental data, but you'll have to listen to his talk to find out more about that because we're running out of time. In the two papers we're talking about today, the data will be obtained via computer simulations using biologically relevant parameters. They don't do the models exactly the same way, but there's a lot of similarity between the two models. So in the 2008 paper, these were the different environments that they looked at. They generated a random walk, they generated the place field locations, and then from that they then outputted spiking data, added some noise, and then they got their spiking data. The 2012 paper will do something similar, but it also wants to take a look at remodeling. So instead of just taking a look at a fixed time where everything is stable, they want to look at the formation. So for example, in this example here, you've got the mouse is moving around in this enclosed environment, and there is this wall here, so the mouse can't go beyond that wall. Note that if you remove the wall, you have a remodeling occurring here, because now we've got a new environment. Now the mouse is in this rectangle instead of the smaller rectangle that it was in before. And you can see there's actually quite a bit of a difference. 2 isn't about the same location, so cell 2 might very well be the same. Cell 2, the place field for cell 2 looks to be about the same for both of them. But for cell 1, it was up here. Now it seems to have moved down here. Cell 4 was here. It's moved up a little bit. Cell 5 has moved down. Cell 3 has moved down. And it's not that the cell is moving, it's the place field corresponding to the cell. So we're drawing the environment, but we do have these 5 cells with their firing rates back in the brain, not moving. But the place fields in the environment that correspond to the brain, there is remapping, and so location of those place fields are different. Any questions about that? So the 2012 paper basically takes a look at the formation of the spatial map using persistent homology. So that's what our 2012 paper out of Gunnar Carlson's group, and Devagyan is one of the speakers in our workshop next week. But in order to generate the data, they both the 2008 and 2012 papers did generate the data, and so they both did very similar things. There were some slight differences. In the 2008 paper, they basically just did a nice random walk around here. In the 2012 paper, they also did a random walk, but they based it it also on how a mouse looks locally. So maybe, a mouse might, peer off into different directions, smell a little bit here, not like it, so smell there, and then maybe go a little bit further. And so it did an even more biologically realistic sampling of the trajectory that a mouse would take through these spaces. You can also see that the environment is slightly different. In this case, they only compared a one hold where the one hold is off-centered, two holes of different sizes, and then also two holes where it was a very narrow space where the mouse could travel. So it's kind of a quasi-linear place, because if the mouse is here, there's really only, two directions it can go in kind of a line. So those are the three spaces that the 2012 paper looked at, as opposed to the 2008 paper looked at these spaces instead. But they are all spaces that you would find in actual biology experiments. They both used the experimental firing rate distributions and experimental place field size in order to generate their computer model to get this firing pattern data. Any questions so far? So in order to compare the assumptions, both papers assumed that the place fields are omnidirectional, so the spiking data did not matter how you traveled through. It did not affect how the firing data was generated. In the 2008 paper, the main difference, as I mentioned before, is that the place fields have been previously formed and are stable. In the 2012 paper, they instead looked at the formation of stable place fields. So eventually they become stable, but we're looking at the formation. How long does it take to form a stable place field? They both, to some degree, take a look at, and covering the entire traversed environment, but if you can tell, this is in a lighter gray, because in the 2012 paper, they actually noticed when you could detect the topology, and it occurred well before the entire space was sampled. So we don't have to sample the entire space, we just need to sample sufficiently to detect the topology. And so you may not even need the majority. So into the 2012, you don't really need the majority, but you do need enough to detect the topology, and we'll show a graph of how much is needed later on. So there also another difference is in the 2008 paper. So in the 2008 paper, they were only interested in H0 and H1. You notice that all the environments were connected. They just varied on the number of holes. So we know what H0 is. H0 should be 1 because it's connected. And then H1 will either be 1 or 2, depending upon whether there's 1 or 2 holes. So that means in order to create the correct topology in the 2012. So in order to create the correct topology in the 2012 paper, I can be, coming along here, and maybe we'll do something like this. But notice I'd have to go through, I meant to actually avoid the pink region. I have to hit the pink region. So if I only hit these regions right here, I get a hole that doesn't exist. And so the path only has to be dense enough to make sure you go through triples if there isn't a hole, and dense enough to actually encircle the holes. So the two things that we need to check is that it needs to be dense enough to go through any triple intersections if it does not correspond to a hole, and dense enough to go around and cover holes. So that was the 2012 paper, just H0 and H1. But it's possible that you might have several triples that might form a tetrahedron. You don't have to be dense enough in 2012 to go through quadruple intersections. You don't have to form the solid tetrahedron because they didn't calculate H2. Versus in the 2008 paper, since they calculated H0 through H4, they needed much more density. So they needed a higher, so they needed more, more coverage in 2008 to get the correct topology than what was required in 2012. And that is for two reasons. The two reasons are, the first one that we mentioned is we only need to recover the topology. We don't need to cover the entire space. And then the second reason is we only need to recover the topology of H0 and H1. In 2012, we're not going to care if H2 and H3 and H4 are not correct, if we don't have all the quadruples and all the quintuples. Any questions there? So we do, however, need holes to, if the holes are larger than the diameters of place fields, that's going to be a problem for both. If you cover your hole, that's a problem for either technique. You do want convexity so that your nerve lemma will hold. And you don't want too much noise, or you're going to have problem with interpreting, for it to be an accurate result. And then there's also the question as to the size and shape of the place fields. In 2008, they used roughly circular, which is a good model in some cases. In 2012, they used ellipsoidal. So there'll be ellipsoidal place fields instead. So that's another difference between the two papers. But the idea is basically the same. You create a random trajectory. You then cover your things with your place fields. And then from each place field, you then create your spiking data. And each of these are created using distributions that are from biological experiments. Questions here? But many of the details, where they were chosen from, the 2 to 3 hertz, that was 2008. They did something different in 2012. So the exact method in which they, chose how to go from, how to create the trajectory, how to create the place cells where they're ellipsoid. So there were some differences in terms of how the spiking data was created. Questions? So, but our still goal is to create our simplicial complex using only our cell groups. But for our cell groups, they are things that co-fire within a specified time period. And both papers use the same time period. So that makes it much easier to compare the two, is they both use the same time period, which is basically 2 theta cycles. If you've ever seen an EEG, you'll notice that it's periodic. So basically they did 2 periods. And so if there was an overlap within 2 periods, then if there was co-firing within those 2 periods, and they looked at offset. So they might have looked at, this time and then offset it a little bit, offset it a little bit. In the 2008 paper, they basically divided it up into 1/5 or 1/8. I don't know if the 2012 specified or if I forgot them, but the time period at least was the same in both of the papers. So now we have our data. So they created this experimental data, and the experimental data is now these how each of the cells fire, and can then determine the co-firing, and from that can create the simplicial complex. And then the question is, do we now have the same topology as the actual environment? Okay. In the 2008 paper, the answer is yes. Even if you have up to 5% noise, you can still, whether you have 1, 2, 3, 4, or 5 holes, so regardless of the number of holes, you still have about, maybe at least 90%, giving you the correct homology for H0 through H4. Even if you go up to 10% noise, you still have more than 50%, of the trials being correct, getting all the HIs correct for I equals to 0 through 4. And so that's not bad with 10% noise or 8% noise. And so it does cover the topology quite well. So the proof of concept worked. The spiking data alone should give you the topology of the environment. Questions there? In the 2012 paper, they were interested in the formation using persistent homology. So what they did is they took a look at how it formed. So you might have certain place cells, and your place cells may or may not intersect depending upon how many place cells you have. And so as you're going through, so the blue one would be the more accurate one, as we go through, we then form the homology. So if we take a look, for example, in this example, maybe we will start here and then go around here, and so a cycle forms. Maybe we then go here, another cycle forms. Oh, now we kill a cycle. And so based upon the time, if we form a cycle, kill the cycle, depending upon where the mouse or rat has traveled. So that is in terms of what we take a look at. So that is what was done in that 2012 paper, where you look at these things forming as the rat is traveling in time. And you can have many cycles forming, but they might then get filled in once you go through the triple intersection. You can also have different components. These things, depending upon the number of cells, you might have things, coming along here, and then maybe it later on goes through here. So eventually the component might go away, but you might actually be having something going through here and then needs to eventually go through another place cell to, close up, to get rid of this, the number of components. You can actually have a number of components depending upon how many place cells you have and corresponding place fields. Questions there? Their main interest was in this T-min, and the T-min corresponds to when the correct topology has been achieved. In this particular example, we're looking at the one with the one hole. For that, we need H0 to be 1. So anytime after this point here, we do have H0 is 1. So we've got this one component here. But we also need H1 to equal to 1 in order to say that we have exactly one hole. And that happens as soon as this hole is filled in. So this hole starts here. As soon as it's filled in, then we have H1 equaling to exactly 1 here. At this stage, it's equal to 3. At this stage, it's 2. At this stage, it's 3. We want our H1 to be 1, and we want it to persist. So we could have had something where, H1 was 1, but then that might have gone away, and then maybe you might have had other sorts of things. So you can have, like here, it looks like we have an H1 equal to 1, but that didn't persist very long, versus this one. This line here is the persistent one corresponding to the actual hole in the environment. So T-min is the main thing in their paper. They're interested in calculating T-min is when you have the persistent bar for H0 and when you have the correct numbers of persistent bars for your H1. and the minimum time when that is achieved, that is what we're interested in. The minimum time when we've got the correct, actually it's the homology, because we're not actually computing the actual topology, but when the homology is correct, we've got the correct H0 and the correct H1, that's our Tmin. So in this case, we detect the topology of the environment in 5.84 minutes. Questions there? Okay. So one can vary the number of cells. One can vary the size of the place field. So you might have something, that's smaller or something that's bigger. You can, vary the shape and the size of the place field. And you can also vary the firing rate. And they actually went all the way up to 40 hertz here. So they basically have these 3 different variables. They did 10 choices for N, S, and F. So they chose 10 numbers between 4 and 400 for the number of cells to check. So this is the number of cells, and you can see there's basically 10 rows corresponding to the number of cells. There's also 10 numbers. They chose 10 numbers between 10 and 90 centimeters to give you the size of the place fields. And they also chose 10 numbers between 2 and 40 hertz to give you the firing rate. So that is 10 to the cubed choices, so that's 1,000 choices. If we fix a particular choice, so maybe N being 200, F max being 10, and size of the place field being 10, we've got that point there. Then for each of these points, so for any coordinate, say 200, 10, 10, for each of the 1,000 points, so we've got 1,000 points within here. For each of those 1,000 points, they then did 10 runs. They used the same, so the mouse's trajectory, they used the same trajectory for each of the 10,000 trials. But what they varied was N, S, and F, but for a fixed N, S, and F, they did 10 runs each. Questions on that? Their graph then is they plotted points, and their graph said that the blue was giving us in terms of, so this one corresponds to the 1 hold, this one corresponds to the 2 hold, and this one corresponds to the 2 hold but nearly linear trajectory. If we take a look at there's 2 things to take a look at. One is the color. The blue is the success rate in the first 25 minutes of the maximum time, and the red is the success required for almost the full time. And so if you take a look at the color of the dot, then it tells you that blue, this was achieved, the majority of the runs occurred in the first 25% of the max time, versus in red, it took you almost the full time to do it. So that's one piece of information in the graph. The other thing, hard to fit everything in here, is the size corresponds to the percentage of correct trials. So remember, they did 10 runs for each of the 10 choices. So it could be that maybe 10 out of the 10 trials are correct, and they used a large circle to denote that 10 out of 10 of them were correct, versus they used a small circle to denote that maybe 2 out of 10 were correct. So 8 out of 10 may not have determined the correct topology, but 2 out of 10 did. So if I take a look at this small blue ball, it tells me 2 things. One of the things it tells me is that maybe only 1 or 2 out of 10 were correct. So that's the size. The blue tells me that that 1 or 2 was achieved within the first 25% of the maximum time. So a lot of information within this three-dimensional graph, but we do have, that's basically the idea. The amount of time it took for the mouse to detect the topology was 4.3 minutes when there was a single hole. So the mouse only needed 4.3 minutes in order to detect the topology. When it was 2 holes, 11.7 minutes were needed to detect the topology. Now remember, that's the maximum time. So this up here is the maximum time that it took to detect the topology. Again, some things didn't detect the topology at all, but those that did took 11.7 minutes for the 2 holes, and then for the almost linear one, it took 9.3 minutes. But you can notice in terms of the color, it might have taken a maximum 9.3 minutes to detect the topology, but most of them detected the topology within the first 25% of the time. Here it was 11.7, but there's a lot of blue in here. Most of them, again, detected within the first 25% of the time. In this first case, now we can understand why this isn't very blue. 4.3 minutes, you know, to detect it within the first 25% of the time, that would have to be very quick. And so here, more things were found within a quicker period of time, but there was a wider distribution. Well, more of the 4.3 minutes was covered. Here, much of the 11.7 minutes was actually obtained within the first 25%, and yeah, there were a few things that it actually did take longer to detect the full topology. Questions there? Being a three-dimensional graph, so first let's also take a look at this figure. This is the percent of the environment covered. Over here is the time in minutes. So you'll note for the 1 hole, that's the blue curve, for the 1 hole, as well as for the 2 holes, so for these 2 configurations, you can see that it covered, about nearly 50% of the environment within the first 5 minutes. And then in, about the 11, nearly 12 minutes, it covered about, 82% of the environment. But notice to get the correct topology, we only needed 4.3 minutes for this one. So we only needed about 4.3 minutes. And so for our blue 1 hole, we needed less than 50%, slightly more than 40% of the environment to be covered. So in terms of the trajectory, we only need to cover about 40% in terms with these place field type stuff. In the 11.7 to detect 2 holes, well, you can imagine detecting 2 holes is harder than detecting 1. And so then, yeah, more of the environment, at least 80% needed to be covered in order to create the correct topology. But we don't need 100% coverage. So that's one thing about the 2012 paper, is since they were looking at the formation, they didn't need 100% coverage. With the green one, that was in about 9.3 minutes, and so you can see, maybe a little bit less than 9.3 minutes, so more time was needed. So not more time, more coverage was needed, but there was also less space to cover, so covering a higher percentage in an almost linear thing also makes sense. So don't need full coverage to detect, depending upon the number of holes. Less than 50% might work, but if you have more holes, then you might need 80% or maybe close to 90%, depending upon the shape and size of your holes. Questions about that? Since it's hard to see graphs that are three-dimensional, one can take slices. And so we can take slices of these things where our mean place field size is 50 centimeters, our mean firing rate is 17 hertz. So if we fix the firing rate, then we have place field size and number of neurons, so we take that slice. Or we can fix the number of cells, take the slice when the number of cells is 325, and take a look at how place field corresponds to firing rate. And you can see, the correct topology is achieved when you've got the big blue dots. And so when we've got the big blue dots, it is achieved relatively quickly. And as long as the dots are big, even if it takes the entire time, we're still getting the correct topology here. And so we still have, with these large dots, the correct topology. And so seeing within the slices might be an easier way to visualize this. Any questions up through here? And one can take a look at additional slices. So this is the mean firing rate. Fix the firing rate at 5, 8, 11, etc. hertz. And so you can take slices and look to see how, if the mean firing rate is very small, then many of your things, you often don't get the correct topology since the dots are so small. But once it gets to certain hertz, then you do start getting the correct topology. If you go too high, you might actually start losing some things. So this one here is actually slightly smaller over there. So too much of a firing rate. If we've got too much of a firing rate, we'll have too many intersections. And if the firing rate goes too high, so that can also be an issue. So low firing rate, then we don't see intersections. Too high a fire rate, we might see too many intersections. And similarly, one can take a look at the place field size ranging from 30, 40, 50. And you can imagine that if your place field size is too small, well, then you're not going to have too much intersection. But if your place field size is too large, you get too much intersection. And if there's a hole someplace, you will not see that hole. So if we cover, this hole with various things because our place fields are too large, then that's also a problem. And so you can see that intermediate values work best. So it tells you basically what is sort of the best place field size in order to correctly identify the topology given by the size of the dot. So here we've correctly identified the topology, and also how quickly you identify the topology, blue being identifying it more quickly than red. So out of our 10 runs, we still got the topology basically identified well here, but not here, but it took us longer with the red dot. Questions? Okay. And one can also take a look at standard deviations in order to find out where is the region stable. So for what values of S max, the size of the place fields, the firing rates, and the number of cells, for what values do you generally get the correct topology? Maybe it will take you longer to get the correct topology, but we just want the big balls, not the small balls. And so one can take a look at the stable learning submanifold, which is basically the best values for S, F, and N that give us the correct topology most of the time out of the 10 runs for a fixed S, F, and N. Any questions about that? I did forget to mention one thing, so let me go back to that slide. Here it is right here. The characteristic, the usual minimal map formation time was about 2 to 5 minutes. For the one hole, your max time was 4.3. For your two holes, the time was larger, but most of the time you made that within the first 25% of the time. Since this is blue, most of the time you detected the topology within the first 25% of the time. And so the usual minimal map formation was between 2 to 5 minutes, which happens to be comparable to the biological learning time. So they know this experimentally, that they actually get a very similar learning time despite the many choices of variables for how the firing data was generated. So, but there seems to be a lot of robustness in this as well. So that's a nice thing that they, got about the same. 2 to 5 minutes is about how long it takes rats to learn, although it can sometimes take longer. Questions there? Okay, so that finishes up the topology. And the 2012 paper was basically concerned just with the topology, but if you remember right, in 2008, we mentioned that we wanted to go ahead and talk about geometry as well. So we can go ahead and also talk about geometry too, using graphs. So we'll be back to the 2008 paper, but I wanted to finish all the topology because the methods used to do the geometry are a bit different than for the topology. So kind of erase how things were created for the topology. They still did do the create a trajectory, then generate place fields, and generate, the place cell firings. So the spike rate, so they generated those. So they still generated those. They did do it slightly differently for determining geometry versus the topology, but the idea behind the method for generating the data was the same. So generating data is the same for the geometry of the topology, with some minor differences in choosing parameters. Questions there? But determining the geometry, they did something quite different. They did not do the simplicial complex that they did before. Instead, they created a graph. And the way they created the graph was to say, well, we'll start off with our vertices corresponding to each of our cells, just like for the simplicial complex. But now we're going to add, instead of edges, we're going to add vertices for each of the intersections. So if we've got an intersection, so cells 1 and cell 2 intersect, we're going to put one here, cells 1 and 3 intersect, 2 and 3 intersect, 1, 2, 3, and 4 intersect. So wherever we've got an intersection, 2 and 4, 2, 3 and 4, 3 and 4, for each of these intersections, we have a vertex. So that's quite different than what we did for the simplicial complex. But the thing is, we want geometry. To get geometry, that means we would like distances. So we'd like to actually create actual distances. So these are the place fields. So we've got our place cells, and that gives us our place fields. We want to look at distances basically between our place fields given the firing data. So again, the idea is the brain only has the firing data. It has to generate the place fields all on its own. But for the geometry, we would like to create distances between place fields, but those distances will be created using only the firing data. Well, one way to do that is basically just to come up with something and see if it works. So what they did is they said that, well, we're just going to say they will renormalize things. So they will say that between any pair of cells, this distance is going to be, not between any pair of cells. Given, so let me erase that. So given a pair of cells that intersect, so if we've got a pair of cells that intersect, we're going to say that this distance right here is 1. It's an arbitrary choice that will then be later renormalized to then compare to, they have the actual place fields, so they can actually measure the actual distance. So this would be, these things here. This is actually, my balls are supposed to be centered around these things. Ignore my lack of artistic stuff. They can actually measure what this distance is between the actual place fields that they have generated, so they actually know what these distances are. But what they'd like to do is go from the spiking data to distances. So one way is we'll just go ahead and say that they are 1. And then we can normalize this so that the mean is 1, so that the place field distance is actually 1. So, or you can normalize to basically the mean for this distance for the place fields. Questions there? What one can then do is basically, if you were to randomly create 2 cells that intersected, if you take a look at just placing a lot of place fields, so not cells but place fields, if you take a number of place fields and took a look at only the ones that intersected, and you looked at the mean distance between the centers of each of these things. So if we took a look at the mean distance here and we calculated that, well, that seems to be on average close to this if you've got k equaling to 2, where we're now interested in now taking a look at this triple intersection right here. Let me clear that so we can take a look at that again. And so we know now the distance between here and here is 1. So between here and here is 1. Between here and here is 1. Between here and here is 1. We now want to add this intersection. And with this intersection, we now have a triple, and that's where k equals to 1 corresponds to when you have an intersection of 2 things. K equals to 2 is when you have an intersection of 3 things. And we now want to say, what should these distances be? And the distances that we're going to have is we're only going to create an edge if the difference in the number of things in the intersection is 1. So since this thing has is the intersection of 3 place fields, we will only draw an edge if the intersection comes from something with 2 place fields. So if we've got an intersection of 2 place fields, we will draw edges to intersections with 3 place fields. If we want to do intersections of 4 place fields, then we would connect them to things that had intersections with 3 place fields. So we only draw an edge, so there's no edge between an intersection of 3 place fields and an intersection of just 1 place field, or an intersection of 5 with an intersection of 2. You only draw an edge when the number of things in your intersection differ by exactly 1. So since we have, this is intersection of 1, 2, and 3, we'll draw an edge to 1, 2, we'll draw an edge to 1, 3, and we'll draw an edge to 2, 3. So that gives us our graph. So we've now identified the graph. In order to do the distances, well, this distance is bound to be shorter than this. You can do a geometric argument, but if you do these things at random, this distance would be shorter. So you want this thing to go decrease, so we do 1 minus this thing. And so that's what their distances are, and they therefore get this graph of distances. And the nice thing about the graph of distances is if you normalize the distance so that the mean distances are so that the mean pairwise distances, so if I take the distance between 2 points over here versus 2 points over here, their mean distance should be about the same for both the simulated data where we have the actual place fields, as well as the calculated data. And so this is the calculated data where they basically took the distances and created a graph that preserves the distances as best as possible. And you can see this is where the original grid where you had the L by L grid that the rat was running in. And so you know what the place fields look like. And you can take a look at if the rat were running along this line, it gets mapped to a line. It's a little bit jagged, but it does basically recreate the structure. It kind of looks rectangular. And so it does do a pretty good job of recreating the distances. And depending upon the number of cells, the mismatch in terms of the pairwise distances is actually, very, very low. And so it does do an excellent job of recreating. Now, it does depend upon the place field radius. You can do different gamma distributions to choose the place field radius. And if the distribution is too wide, then your error is not so good. But if you have a nice distribution, then you have a pretty low error. And one does have to do a little bit in order to get this thing to match up with this, because if you're only looking to preserve the distances, this map is just as good as this, but then one can do translations to kind of map these things out. The last thing in the remaining -1 minute is that this also gives us a way to do the multiplicate fields. So before, we have to be able to determine that this field and this field are different. If you've got a cell that signals when you're here as well as when you're here, when you travel through, we need to be able to distinguish between these two cells. And you can use the geometry to do that, and thus you can use that to get your correct topology, even if 10% of your cells have two different are multi-peaked, have two place fields, so they might have two place fields that correspond. Even if 10% of your cells have that, you can still get a pretty good match of your topology. So the geometry can actually play back into the topology. Okay. So sorry for going over today. Any questions anyway? So then I will see you on Friday.